Hello, in this video we will look at the IMP synthesis. This is a biochemistry lesson we will discuss, and we will discuss about the whole process. So first of all we will understand about from the sucrose. So the sucrose is the type of a disaccharide which that is come from the sugarcane, for example, that convert into the glucose, and it is also come from the starch that is convert into the glucose. And after this, glucose 6 phosphate convert into the glycolysis, and that's NADH produced, and this is the 6 glucose phosphate will be produced. And again NADH produced and carbon dioxide released to produce the ribulose 5 phosphate. So this is the ribose 5 phosphate is the basically PRPP is the pyrophosphate, that is the 5 phosphoribosyl uh, uh, pyrophosphate that is formed. So in this way we will discuss about the from alpha D ribose 5 phosphate, which that is convert into the 5 phosphoribosyl um, pyrophosphate. So this is the PRPP double P pyrophosphate. So the ATP used to convert AMP and the 2 phosphate group will give to the alpha D ribose 5 phosphate in the presence of ribose phosphate pyrophosphokinase enzyme. And in this way the 5 phosphoribosyl X pyrophosphate will be formed. This is known as the PRPP. So in this way, another is the further processes will be in the presence of glutamine, and water will use in the presence of amidophosphorybosyl transferase enzyme to release the and separate the glutamate, and the pyrophosphate will be separate and to form a step 3 is the basically yes, this is the 3,5 phosphoribosyl amine. This is the PRA, and this is formed, and this is basically is 3,5 phosphoribosyl formation. After this formation, it is donation of the amine group in the carbon number 1, and after the glycine and ATP used in the presence of the glycine ribosyl synthase enzyme used to separate the ADP and phosphate group. So that is mean the energy requiring step. And in this way, the glycine amide ribosyl 5 phosphate will be formed, GRP. And in this way, you can see in the figure. And another thing is the GAR transformylase enzyme use, and the N10 formyl THF is used for it, and that's release the THF, and the formyl will be N10 is the formyl which that is attached. You can see in this diagram, and this formyl glycinamide ribotide formation, formyl glycinamide ribotide formation will be occur. And after this, FGAM synthase enzyme use in the presence of water and glutamine as well as ATP. After this, ATP and water and glutamine used to separate the glutamate, and in this way you can see in this diagram that is the basically is the production of here is the ribose phosphate and formyl glycinamide ribotide formation, that is known as the FGAM. So this is the ribose phosphate formation. Here is the ribose phosphate, but the additional compound is known as the formyl glycinamide. And this is the formyl glycinamide will convert in the presence of ATP to release the ADP. It means the energy requiring step, and the FAGM dehydrogenase enzyme used, formyl glycinamide ribotide glycinamide dehydrogenase, sorry. So this convert an NADH dehydrogenase, it means the NADH will be used. NAD plus, here you can see this is the separation of the hydrogen, is the proton, and accept by the NAD plus to 2 NADH form due to the 2 hydrogen release from it, from this reaction. So you can see in this diagram. So the next step is the formation of 5 amino imidazole ribotide formation. So the ribotide means the ribose 5 phosphate is in the form of ribotide. So here is the carbon which that is left behind, and oxygen is released also. It can be converted by the dehydrogenase enzyme to release water. So that is important things, and it can be released in the form of water or the NADH formation. So important is the true thing is that the water will be released. So that is why the oxygen will also remove. So on the other hand, here is you can see the carbon dioxide and the AIR, which that is the AIR carboxylase enzyme used, and here is you can see that formation is a carboxy aminoimidazole ribosyl 5 phosphate formation. And in this way the hydrogen will be accepted by the NAD plus, and the carbon dioxide will be bind with this carboxy amino imidazole formation. So you can see that is the carboxylation will be occur. So the ATP used, so it is the energy requiring step. And here you can see the aspartate and ATP use, and this will be released into the ADP, and that is the NADH is also produced, and this is synthase enzyme used for it to produce the 5 amino imidazole succinyl carboxyamide ribosyl 5 phosphate, and this is the structure is looking like this. And in this way the addition of the here you can see the aspartate addition. So this is the aspartate, and here you can see this is the aspartate will be add. So the oxygen is the same ratio, and here is the addition of the nitrogen aspartate, nitrogen containing the bond will be occur. So in this way the bonding basically is the bonding with this here is, and here is the 5 carbon containing also the nitrogen. So that is why it is known as the 5 amino imidazole succinyl carboxymide ribosyl 5 phosphate, because of 5 carbon containing phosphate group also. So on the other hand, here is the fumarate will be separate in the presence of adenosyl succinate lyase enzyme. This adenosyl succinate lyase enzyme will form the, after the separation of the fumarate, will form the 5 amino imidazole 4 carboxyamide ribotide formation. So this fumarate, what is the benefit of the fumarate? Fumarate is used with the binding with the water to produce malate, and this malate in the presence of NAD plus dehydrogenation to produce oxaloacetate, and this oxaloacetate will bind with the acetyl CoA to go into the Krebs cycle to produce more energy. So that is the energy is required for this step, so that's energy recovery will be occur. So on the other hand, important thing is that here is a 5 amino imidazole 4 carboxyamide ribotide formation will lead to further changes. So here you can see that is the phosphate group attached with the ribose, and this here is the ring formation will begin. In this way, so the N10 formyl THF is used to release in the form of THF, and here is the alka transformylase enzyme used, alka transformylase enzyme, ALCAR transformylase enzyme basically, which that is form. Here is the ring formation. So this ring formation will known as the ribose 5 phosphate bind with the ring is known as the 5-farmino uh, imidazole 4-carboxyamide ribotide. So this is basically, here you can see, and this is in the form of IMP cyclohydrolase enzyme to release the water. And here you can see the ring formation will be occur, and this ring formation is a complete IMP. So here is the binding with the, here you can see this is the ring formation will be occur after the separation of the water. So this is the IMP. It is known as the inosine monophosphate. And this inosine monophosphate is, I am drawing here again to understand for the further process. And what is the benefit of the IMP? So that was the that is the IMP is used. Here is the nitrogenous base, and here is the phosphate group, and this is the ribose sugar, and it means it is the basically the precursor of something we will discuss. IMP is the precursor of different molecules like cyclic AMP, IDP, ATP, GTP, AMP, second messenger formation, as well as the DNA synthesis. So this all thing is in the form of, this is the nucleotide. So this is nucleotide is used for the synthesis of DNA, new DNA during cell division, while due to the replication of the DNA, which that is bind the nucleotide. So the different nucleotide formation we will not understand, we will just understand about the further processes. So IMP dehydrogenase enzyme also use, and NAD plus and water will add to release the NADH, and in this form this will be produced. And on the other hand, the adenylosuccinate synthetase enzyme used, and the GTP and aspartate will be really used to another way to produce the adenosyl succinate lyase enzyme production. So here is the another thing is that the XMP will be formed. So this is the XMP is the GMP synthetase enzyme used to ATP and glutamate and water used to release the AMP and pyrophosphate and glutamate. So this formation is known as the GMP, the guanosine monophosphate. So this nitrogen base is the guanosine. So here is the guanosine monophosphate formation, and the phosphorylation to become a GDP, and this GDP will convert in the Krebs cycle to convert the GTP, and this GTP is come from the phosphate group, come from the ATP. So the ATP will be formed. So this GDP basically convert into the GTP in the Krebs cycle, and after this the GTP is donation of the phosphate group to the ATP. So that is the important things. On the other hand, here important thing is that the adenylosuccinate lyase enzyme formation. So this enzyme formation will further process and release the fumarate. This fumarate again used for the Krebs cycle. So this is the AMP and cyclic AMP formation, the second messenger in intracellular, and the ADP formation and ATP are used as an energy in the Krebs cycle, as well as the glycolysis and the electron transport chain. So this all thing is done here, which that is the DNA formation and energy, as well as the messengers for messages, molecular message. So thanks for watching.